All right, welcome everybody. Thanks so much for coming to the LEAP lecture today. Also, welcome to those online who we can't see in the room, but welcome. So it is my great pleasure to introduce Mary Margaret Stoll here, who's our lecturer. Mary Margaret got her PhD just a few months ago at UW in oceanography, where she worked on a real variety of things, including the use of geochemical proxies to understand past ocean acidification, looking at corals in the coastal California area. In fact, she had a paper published less than a week ago in Nature Communications on that. Looking at a 130-year reconstruction of ocean acidification, which is really important for understanding how species have evolved and changed. Of course, 130 years is a lot longer than we've directly been observing ocean carbon chemistry and its change. She's also worked with models in this work, and collaborated with lots of different people, and also looked at marine carbon dioxide removal questions, and particularly ocean alkalinity enhancement, which has a big effort going on there in the Seattle area. And then she's also extended her work into machine learning, so which is of course the topic of today's talk, which is going to be talking about leveraging four-dimensionally mapped ocean biogeochemistry to inform species distribution modeling. So we're really happy to have you here. And yes, and Mary Margaret is staying in Seattle as a postdoc starting in January at PMEL and UW. So yeah, let's welcome Mary Margaret and look forward to your talk.
Thank you. Great. Well, thank you for the introduction, and thank you all so much for being here. As Galen said, my name is Mary Margaret Stoll, and I recently defended my PhD in oceanography from the University of Washington. Still feels pretty surreal to say that, and I was realizing as coming over here today, this is the first presentation that I've given since my defense, so I have like some post-residual jitters kind of coming off of that still. But I'm really excited to be here at Columbia today, and get to see the LEAP Center in person. I've kind of been following LEAP from afar for a while now, and it's clear there's some really important and impactful research happening here. So looking forward to learning more from all of you today and chatting with many of you at lunch. So today I will be talking about bridging ocean biogeochemistry and fisheries by utilizing new biogeochemical data products and using machine learning. So I will say up front, this talk is probably a little bit different from the typical topics that you see here. This is certainly a more applied use case of machine learning and climate and ocean data, but hopefully I can bring something new and interesting to all of you. So first off, I want to thank all of my co-authors on this project and my advisor, Andrea Fassbender at NOAA PMEL. This was a really interdisciplinary project, so kind of provided the opportunity to bring together a large team of wonderful people, some with expertise in fisheries, others in BGC-Argo, and others in machine learning. So huge thank you to my team of co-authors for their support of this work. Now, before I kind of jump into some of the research, I just wanted to give a quick background introduction of myself. So I'm actually from the East Coast of the US. I'm looping this talk into travel home for Thanksgiving. So it's worked out very well for me. I went to undergrad at Amherst College, had a really hard time picking a major. I knew I wanted to study a STEM field, but really had a hard time deciding which one. I dabbled in engineering for a little bit at Dartmouth College, but eventually was drawn to oceanography because it brings together all of the scientific disciplines. You really have to know a little bit of the chemistry and the physics and the biology to even begin to grasp the Earth system as a whole. So I pursued a PhD in oceanography at the University of Washington. I was told that many of you might not be coming from an ocean background, so I've taken it upon myself to share some of the major selling points of oceanography. One of my favorites being that it brings you to some pretty incredible places. I've had the opportunity to do field work from French Polynesia to Alaska, and even in my backyard, the coastal waters of Washington. So I would highly recommend getting on a boat if you ever have the opportunity to do so. Oceanography is awesome. I feel really grateful to have been given all of these opportunities, though it certainly wasn't always in a straight line. So happy to chat more about that as well, if folks are interested. So Galen kind of alluded to this a little bit, but over the course of my PhD at UW, I had the opportunity to work across a surprisingly diverse set of topics, from reconstructing past ocean acidification using paleoproxies to kind of determining the detectability of marine carbon dioxide removal to modeling distributions of fisheries. So I kind of like to joke that one of the hardest parts of my PhD was actually writing the introduction of my dissertation, because it's not completely obvious how all of these topics tie together, but they really are all connected under an umbrella of using new biogeochemical tools and observations and models to understand our changing ocean and how those changes will impact both society and ecosystems. So again, for today's talk, I'm only going to focus on this final project in graduate school, which kind of assessed the value of biogeochemical information in modeling the distribution of a fisheries species. That's kind of the only project in grad school that I used kind of machine learning heavily. So I'll start with a pretty broad overview, quick overview of ocean biogeochemistry for those of you who are not as familiar, and why these chemical and biological processes and cycles are essential to marine ecosystems. So the oceans, of course, play a really central role in the Earth system. It's an incredibly dynamic place. I love this animation from NASA. Circulates on many physical scales and also connects tightly with the atmosphere and the land. So any changes in the ocean really ripple through the Earth system. And at the core of this interconnected system are the ocean's biogeochemical cycles, including carbon, nutrients like nitrate and phosphate, and oxygen. So these cycles impact ocean characteristics such as its chemistry and acidity. They sustain marine ecosystems, they regulate Earth's climate, and these photosynthetic plankton are responsible for about half of the world's oxygen. So these cycles are really critical and essential to life as we know it. All of us here know that anthropogenic climate change is driving a suite of changes in the ocean. The ocean has absorbed about a quarter of anthropogenic CO2 emissions, causing seawater to acidify. Warmer water holds less oxygen, so low oxygen zones in the ocean are expanding and compressing suitable habitat for marine organisms. So all of these changes ultimately impact marine ecosystems and organisms and the people who rely on a thriving ocean. So the biogeochemical landscape in the ocean imposes really critical constraints on marine habitat. So low oxygen levels can cause hypoxia, and high PCO2, high carbon dioxide in the ocean, can cause hypercapnia. Both of these processes can impact metabolic rates of organisms and increase or impact stress tolerances for organisms. Different marine organisms have different temperature thresholds, and of course, biological productivity in the ocean is a really important proxy for optimal foraging conditions. So the biogeochemical characteristics of the ocean really bound suitable habitat today and will also govern how species movements and behavior will change in the face of climate change. So marine organisms are kind of constantly navigating this shifting landscape of food availability and temperature, oxygen, and acidity thresholds. And to capture that complexity, or To translate those environmental conditions into spatial predictions of suitable habitat, we turn to a type of model called species distribution models, or SDMs. These SDMs are models that predict the distribution of a species across space and time. They can kind of produce maps of suitable habitat or population distributions. They're used for a really wide range of purposes, including avoiding protected species bycatch, identifying regions for conservation, and projecting kind of large-scale ecosystem shifts in the face of climate change. So these models are incredibly versatile and used across marine science and management. They're also quite instrumental in the world of fisheries and fisheries management more specifically. Fisheries, biologically, culturally, economically important. In the United States alone, they contribute over 1.5 million jobs and billions of dollars in sales added to the economy. Worldwide, over 3 billion people rely on fisheries and other seafoods for nutrition and protein. So understanding the role that biogeochemistry plays in influencing these species movements and how these relationships will change in a changing ocean is really, really important. Traditionally, species distribution models have relied heavily on surface variables like sea surface temperature and surface chlorophyll. But as we've discussed, the conditions that bound suitable habitat often occur below the surface and across multiple dimensions of ocean biogeochemistry. So these characteristics often cannot be observed with satellites alone. That kind of brings me to the goal of my research, which is to understand the value of subsurface biogeochemical information in predicting species habitat use. And this information, we think, may produce more reliable mechanistic relationships between species and their environment. So I mentioned again, traditional observations of ocean biogeochemistry have relied heavily on satellites and ships, but those observations are either limited to the surface ocean or have very sparse spatial coverage. But the Biogeochemical Argo float array, which is composed of autonomous profiling floats, has really transformed our view of the subsurface ocean in both space and time, and really enabled the study of biogeochemical processes from a new perspective. So these Biogeochemical Argo floats, or BGC-Argo floats, I'll call them, are equipped to measure a variety of biogeochemical characteristics, including pH, oxygen, nitrate, biooptics. And the robust distribution of these observations has facilitated the creation of these 4-dimensionally mapped data products. So these time-resolved, depth-resolved data products really mark a notable shift in our ability to investigate fundamental oceanography and biogeochemical processes concerning productivity, deoxygenation, and marine ecosystem health. So there are really countless ways to leverage these data products to study fundamental oceanography, but today I'll be sharing kind of an application to the world of fisheries. So my research really focuses on the ways in which these 4D biogeochemistry data products can be leveraged to characterize the environmental conditions experienced by marine species and in turn inform a fisheries species distribution model. To do this, we used juvenile Pacific albacore tuna as a case study organism, given their highly migratory nature and their economic value as a US fishery. These albacore can be classified into 3 migratory subgroups, including long migratory, short migratory, and resident fish. But today I'm primarily going to focus on this long migratory class. So these long migratory albacore undertake really extensive seasonal migrations between the subtropical North Pacific in the winter and spring, and the temperate coastal ecosystems of the California current during the summer and fall. In addition to their really expansive horizontal range, they also exhibit energetic diving behavior, so frequently diving to depths beyond 200 meters during the day. I actually recently just read a paper that said in a single day, a juvenile albacore tuna can travel upwards of 6 miles just in vertical travel in a single day. So these are, the endurance is quite impressive for these fish. But the horizontal and the vertical range of these fish cause the species to encounter a really broad range of environmental regimes, each with very distinct biogeochemical characteristics. So this really makes for a fantastic case study organism to kind of explore the value of biogeochemical information in modeling their distributions and migratory behavior. So to evaluate how this 4D biogeochemical information might inform albacore habitat use, we built machine learning classification models that predict the probability of a fish presence or absence based on some environmental characteristics. So this slide kind of summarizes the main methods and data that we use in this study, but I'll kind of walk through each component in a little bit more detail over the next few slides, so sit tight. So each model uses a subset of environmental predictors, including temperature, oxygen, and these proxies for optimal foraging conditions, chlorophyll and particulate organic carbon, or POC. We use the XGBoost classifier as our machine learning algorithm, and then the model is trained to predict fish presence or absence, or kind of the probability of fish occurrence using these environmental predictors. I also want to note, for the purposes of this study, when I say biogeochemical information, I'm specifically talking about these 3 variables. We already know that temperature is a very strong predictor of fish movements from a physiological standpoint. So we're really interested in determining where and when and how biogeochemistry kind of modulates their habitat use. Just a quick few notes on model construction for those of you who are more machine learning inclined, which is maybe many of you. So I mentioned that we used XGBoost as our machine learning algorithm. This is a decision tree-based ensemble learning method that's particularly known for being computationally efficient and skillful. It builds trees sequentially as opposed to in parallel, and uses a gradient descent algorithm to minimize some loss function. As for the model construction, we used a 5-fold cross-validation design using a leave fish out technique, and we did that to kind of maintain independence between our fish samples. So each split here has the same distribution of all 3 of our migratory classes: the long migratory, short migratory, and resident fish. We then assessed the skill of the models using a few different evaluation metrics. We calculated the validation accuracy, the F1 score, which is just a metric that weights precision and recall, and AUC-ROC, the area under the receiver operating characteristic curve. This is a very common metric for binary classification, kind of produces a curve that assesses the balance between the true positive rate and the false positive rate at different thresholds. But for all of these metrics, values closer to 1 are better. So if you remember nothing else, remember that. I also just wanted to share some brief information on the data sets that we use. So the albacore tuna data comes from 2 different tagging programs that tag and release juvenile fish. And these are tiny tags that are attached to the fish, and they have the ability to record and store information like ambient temperature, body temperature, location, and dive depth of the fish. So you might be wondering how these tags are retrieved, and I too was curious about this. And it turns out that these programs basically post a bunch of wanted signs for these fish at marinas across the west coast of the US, which I find pretty hilarious. So, these signs provide information on like the catch reward for catching one of these fish and how to remove the tags, where to return the tags, et cetera. So we have a really rich data set of presences across space and time spanning almost 10 years. So here we're looking at a heat map of presences where the darker red colors indicate either multiple fish in that grid cell or a single fish spent multiple days in that grid cell. Now, if a model is to predict a fish presence or an absence, we also have to teach the model where the fish did not go. And since it's not customary for fisher people to report where they went fishing but returned empty-handed, we have to generate this absences data set ourselves. And these are called pseudo-absences. There are a number of methods used in species distribution modeling to generate this type of data set, but for the purposes of our study, we just use background random sampling from the domain. So our machine learning model was trained on a balanced data set of presences and this generated data set of pseudo-absences. All of the biogeochemical data products that we use were informed by Argo or BGC-Argo float data. So we used two depth-resolved data products of POC and chlorophyll, again, our foraging proxies from CMIMS, the Copernicus Marine Service. And these two products use something called the SOCA method, which merges satellite ocean color with Argo data to infer the vertical distribution of a given variable of interest, in this case, POC and chlorophyll. So this method uses several different variables from BGC-Argo and satellites as inputs to a neural network. And the downstream result is a gridded data set for POC and chlorophyll that is both time and depth resolved. The oxygen data product that we use is called GoBio2. It's also time resolved, depth resolved, machine learning based product that has monthly resolution. I believe it spans 2002 to, I think it's updated to 2024 now, or 2023 maybe. And the temperature data product is derived from Romick and Gilson 2009. Again, monthly resolution, depth resolved, I believe, to 2,000 meters. But that's far deeper than our fish care to venture. So those are kind of the main tools and methods that we used to explore the value of this depth-resolved biogeochemical information in our fisheries species distribution model. So with that background, I want to introduce the two main research questions that we aimed to address. So first, we were interested in exploring the value of subsurface information in predicting albacore habitat use. And to do this, we created two-dimensional species distribution models, so something that looks like this. So the model predicts the probability of fish occurrence in latitude, longitude, and time. And the two species distribution models are informed by different data products. One of them is trained on surface satellite information. The other is trained on depth integrated data from the float, so has some subsurface information. So comparing the performance between these two models allows us to kind of evaluate the value, or evaluate how subsurface biogeochemistry influences albacore movements. Next, we were interested in determining how well we can predict albacore movements in three spatial dimensions. And to do this, we created a three-dimensional species distribution model that predicts the probability of occurrence in latitude, longitude, time, and depth now. So this kind of 3D probability of occurrence model. And we can also analyze the influence of different biogeochemical predictors on suitable habitat with this model. Okay, so moving on to some results. So again, our first question addresses the value of subsurface biogeochemical information in predicting albacore habitat use. So I mentioned that we compared two different species distribution models, one informed by surface satellite information, the other informed by depth integrated kind of float-based data. And by depth integrated, I mean that we take a variable like chlorophyll and integrate it to some prescribed depth. So it essentially collapses kind of the vertical structure of that variable into a single representative value for each grid cell. So this new parameter kind of reflects conditions throughout the water column, not just at the surface. So again, while surface conditions can sometimes serve as a proxy for subsurface conditions, that's not always the case. We absolutely see instances where surface and subsurface conditions decouple from one another, and that information is not captured by satellite. So here is just one example of several fish tracks from the month of June overlaid on maps of surface chlorophyll observed from satellites on the left and depth integrated chlorophyll, again, informed by floats on the right. So we see that integration with depth really changes the patterns of chlorophyll hotspots that we see. So we're really interested in understanding how fish migration patterns might be driven by these subsurface signals that satellites can't capture. So we compared the performance of these two different models, which were again trained on similar but slightly different environmental predictors. The satellite model was trained on SST, sea surface temperature, and surface satellite chlorophyll and POC, while the float model was trained on sea surface temperature and depth integrated chlorophyll and POC. Okay, so first taking a look at habitat predictions from our float model, the model informed by depth integrated variables. So these plots are showing modeled mean probability of fish occurrence by season. So red colors indicate a higher probability of a fish presence. It kind of serves as a proxy for suitable habitat, whereas blue colors indicate a lower probability of occurrence. In general, the float model kind of captures general migration patterns that we see in the fish tagging data. In general, the fish reside in the subtropical North Pacific during the winter and spring, and then migrate eastward to the California current during the summer and the fall. Our global evaluation metrics also demonstrate this model is fairly skillful. Again, values closer to 1 indicate a more skillful model, especially considering that we're using just 3 predictor variables here. We were next interested in assessing the performance of our satellite model against the float-based model. And we found, in general, the float model outperformed the satellite model when comparing the global evaluation metrics, but the value of the float model was particularly pronounced in specific regions and specific seasons. So for this demonstration, I'm just going to focus on one season in the spring. So here we're looking at modeled probabilities of occurrence in the spring from the float model on the left, satellite model on the right. In general, we see fairly similar patterns of habitat suitability, but divergences between the two models becomes a lot more clear when we take the difference between these two probability predictions. So here we're looking at the difference in predictions between the two models. And in this, the orange colors here indicate that the float model predicted a higher probability of fish occurrence or presence in that region. And we see in the spring, the float model predicts higher probability of occurrence or fish presences in this kind of northeastern subtropical Pacific region compared to the satellite model, which is kind of interesting. But what we really want to know is if this increased probability of finding a fish suggested by the float model is actually accurate. Does that align with behaviors that we are seeing with the fish? So if we overlay the locations where only the float model accurately classified a fish presence, we see that it overlaps quite nicely with the region where the float model predicted a higher probability of presences. So all of these circled locations are presences that the satellite model incorrectly classified. We conducted a kind of regional performance evaluation in this area, and the float-based model showed a 25% improvement in classification accuracy, so identifying fish presences compared to the satellite model. So we were next interested in trying to understand why the float model was particularly skillful in this region, why it outperformed the satellite model by so much. And to do this, we compared surface satellite chlorophyll with depth integrated chlorophyll through time. So we calculated kind of a correlation at each grid cell through time. So in this map, red indicates a strong positive correlation between surface and depth integrated values, and blue indicates a negative correlation. And again, these black circles mark locations where only the float model correctly identified a fish presence. And at those locations, we see little or weak negative correlations for chlorophyll, meaning that surface conditions are not a reliable proxy for subsurface conditions in this area. So this decoupling kind of helps to explain why the satellite model may have missed some suitable habitat here that the fish are actually using. We then looked at fish diving behavior in this region. Since only the float model captured the true presences here, we wanted to understand whether those fish were diving differently in a way that might explain the float model's improved performance in this area. So on the right here, we're comparing violin plots of dive depths for fish that were classified correctly by each model. So we see that the float model in purple on the left classified presences where the fish dove significantly deeper in the water column compared to the satellite model. So in general, the float model identified presences where the fish dove on average around 100 meters in the water column, whereas the satellite model identified presences where the fish were diving only to about 50 or so meters. So this pattern kind of supports the idea that subsurface biogeochemical information can help explain habitat use and behavior in this region, and that these conditions aren't visible to satellites, which is why the float model performed much better in this particular area. So just taking a step back for a second to kind of summarize some important points so far. This first research question addressed the value of subsurface information in predicting albacore habitat use. And to do this, we created two different species distribution models, one informed by surface satellite information, the other informed by depth integrated data from floats. And we found that subsurface information improved predictions of habitat suitability, especially in particular regions and seasons where we see decoupling between surface and subsurface conditions. We also found that the float model was able to accurately classify presences when the fish dive much deeper in the water column. So this kind of indicates that subsurface information is helping to constrain habitat use, especially when that information isn't readily obvious from surface satellite information. So from here, we created a three-dimensional species distribution model that, again, predicts suitable habitat in latitude, longitude, time, and depth to understand how this biogeochemical information is shaping albacore habitat use in three spatial dimensions. Okay, so we created this 3D species distribution model, which was trained on sea surface temperature and depth resolved chlorophyll, POC, temperature, and oxygen. So here we are looking at modeled probabilities of occurrence in winter on the left and summer on the right. Again, red colors indicate a higher probability of fish presence, which kind of serves as a proxy for optimal albacore habitat. Again, some notable patterns emerge here that also align with observations from the fish data. So again, fish migrate westward in the winter, tend to reside in the subtropical North Pacific, whereas they reside along the coast in kind of the temperate ecosystems of the California current during the summer. We also see that the model suggests that the fish dive much deeper in the water column in the winter, whereas the fish tend to reside in kind of the upper water column along the coast. And that also aligns with behaviors that we see in the fish tagging data. Again, just quickly showing some of these evaluation metrics. Again, values closer to 1 here indicate a more skillful model. So again, a fairly high-performing model, especially considering we really aren't using too many predictor variables here. So we can further understand this 3D habitat use by looking at a time series of fish diving depth and modeled predictions of occurrence along the fish track. So here we're looking at mean daily dive depths of a single long migratory fish over the course of almost 2 years. And again, we see this pattern where the fish dive deeper in the subtropical North Pacific, whereas they tend to reside in the upper water column in the California current. We can plot the modeled predictions of probability behind this fish track, and we see that the model does a pretty darn good job of predicting where, on average, the fish tends to reside in the water column. Mind you, this fish track was left out of the training of this model. The model has not seen this specific fish track before. We can then decompose these predictions into different combinations of predictor variables. So, for example, we can ask, how does temperature alone constrain vertical habitat use along this fish track? And we can kind of take this one step further to understand where and how biogeochemical information is influencing fish behavior by simply taking the difference between these 2 plots. So again, we know that temperature is a very strong predictor of fish movements from a physiological standpoint, but we're really interested in isolating the role of biogeochemistry, so chlorophyll, POC, and oxygen, to determine where and how these variables are influencing habitat use throughout the water column. So taking a look at the difference in probabilities of occurrence between the 2 model configurations that we saw on the previous slide, we can see where these biogeochemical predictors either increase or decrease the likelihood or probability of occurrence along the fish track. So here, brown colors show areas where biogeochemistry decreased the likelihood of a fish presence, whereas green colors show areas where biogeochemistry increased the likelihood of a fish presence. And what's really interesting is this pattern of biogeochemistry either contracting or kind of expanding habitat is consistent across all of our long migratory fish tracks. And this pattern is also linked to distinct regions, distinct biogeographic regimes. So if we sync this kind of time series above with the swimming trajectory of the same fish, we can see kind of where these green and brown colors align in different regions and times of the year. So the brown colors, which again indicate a contraction of habitat, tend to appear in the subtropical North Pacific. So this indicates that albacore avoided deeper dive depths due to suboptimal biogeochemical conditions, even though temperature was well within their tolerance range. On the other hand, the green colors tend to appear in the California current region, indicating that biogeochemical variables actually expanded suitable habitat at depth. So this likely reflects improved foraging opportunities at depth, even though temperatures were less than optimal. So we kind of scaled this analysis up to the entire North Pacific, and we found that the inclusion of these biogeochemical variables contracted suitable habitat by over 60% in the winter and expanded suitable habitat in the summer by 25%. And again, a vast majority of this contraction and expansion is occurring with respect to the depth dimension. So this perspective and understanding was really enabled by these four-dimensional data products. So that was really exciting to see. But these biogeochemical variables are playing a huge role in shaping habitat use and really can provide some new insights when modeling species distributions. Now, to this point, so far, we've kind of been probing the combined impact of our biogeochemical variables, so kind of the collective impact of oxygen, chlorophyll, and POC together. We can use other tools like feature importance to understand where and when each variable contributes more to predictions of suitable habitat. I do want to note some of these predictors are correlated despite selecting variables to minimize correlation. So these feature importances are very coarse, but they still offer kind of a useful first look at how the model is using each variable. So here we are looking at the relative importance of each predictor across the entire domain and all time. So the sum of each of these bars totals to 1. In general, the higher the bar, the more important the predictor. And we see that sea surface temperature and depth resolved temperature are kind of some of the most important features. Unsurprisingly, again, temperature imposes a really important physiological constraint on fish movements. But we also see that the importance of these variables changes quite a bit with season. So we see that biogeochemical variables collectively are the most predictive in the summer. Again, this aligns when the fish largely reside in the California current. And this is also a season when upwelling is the strongest. Productivity is kind of flourishing in this region at that time. Another thing that kind of stood out to me looking at these plots was the increase in the importance of oxygen in the fall. So again, that's largely when the fish reside in the California current region. And oxygen actually becomes the second most important predictor, surpassing depth resolved temperature, kind of accounts for like a quarter of feature importance. So I don't have time to dig into all of the analyses concerning some of these plots, but I did want to show just a brief slide or two from kind of a case study that we did in the California current region in the fall, where we see this increase in the importance of oxygen. So to understand the influence of oxygen in the California current during this fall season, we again ran our model with 2 different configurations of predictor variables to kind of extract the influence of oxygen on these predictions of suitable habitat. So we took the difference between 2 models trained with and without oxygen. So here we're kind of looking at a 3D plot of the difference in mean probabilities of occurrence. And in general, we see blue colors here, which indicates that the inclusion of oxygen decreased the likelihood of a presence, or kind of decreased the suitability of that habitat. And in fact, the inclusion of oxygen in this model for this time of the year contracted habitat by about 15 to 20% in the California current region during these fall months. And again, especially at depth in the water column. So we next looked at oxygen data in the California current to kind of mechanistically explain this decrease in suitable habitat that is seemingly attributed to oxygen. And we actually find that oxygen in the upper 150 meters of the water column reaches its lowest levels in the California current during October and November. So here we're looking at kind of again, a 3D slice of subsurface oxygen along the west coast of the US. And the red colors indicate the threshold value of oxygen that's been cited for albacore tuna. So these low oxygen levels shoal in the fall begin to encroach on what would otherwise be suitable habitat for these fish. So this is a very quick, fairly simple example just to show the ways that we can use these tools to kind of disentangle drivers of species movements across space and time. But there are really kind of countless other case studies that can be used to understand species movements and migratory behavior within their environmental context. So I'll just wrap up here with a few concluding thoughts. So in this study, we leveraged archival tagging datasets, these new mapped ocean biogeochemistry data products informed by BGC-Argo and machine learning methods to evaluate how this information influences the habitat of albacore tuna. And this work really kind of advances the field of species distribution modeling by demonstrating the value of these new data products for the first time. And really at the heart of this advancement is the BGC-Argo float array and the way that it's enabled new insights into the linkages between species distributions and their environmental context. So continuation of these types of advances in species distribution modeling really depends on the persistence and growth of the Argo float array. But again, the framework that we presented here has broad applications across contexts in the marine environment, anywhere from improving bycatch avoidance to informing fisheries management and conservation. So again, there are really kind of endless questions that can be answered with this framework, kind of bridging ocean biogeochemistry and fisheries with machine learning and applying BGC-Argo to ecological questions. So that concludes my presentation. I thank you again for this opportunity to speak with you all. It's been such a pleasure and looking forward to chatting with you after this.
Thanks so much, Mary Margaret. Are there any questions for Mary Margaret? Yeah, Viviana.
Thanks, Mary Margaret. That was like a really super interesting talk. I have a million questions, but I'll start with one, maybe we'll
Happy to
chat.
chat.
One of the things like it's such a cool idea, right? The tagging of the fish. I think for those of us who work in machine learning, this is usually not how you collect data. So it was like, very, very cool to see. I'm sure you guys have thought about this. How, do you expect your data to be reasonably complete? Do you feel like that, it's possible that you are missing fish in some regions, in some conditions? Will this matter? Like, yeah, I don't know. Maybe this is the absolute best one can do, but I'm sure you've spent time thinking about this. So I'm curious to hear.
Yes, absolutely. That's a really great question. I will also caveat, I am not a fisheries person. I'm one of the biogeochemistry people, so take everything I say with a grain of salt. But this tagging program for albacore tuna is relatively robust compared to other marine species. So, for this studies, we used about 30 tagged fish, ranging in time from 6 months to 2 or 3 years. So that's relatively robust for this type of study. But with that being said, there are, we can really only capture the type of behavior from the fish that we are tagging. And I think that in general, the data set that we have here is fairly representative of the fish that migrate from the west coast of the US. But again, there are other North Pacific albacore tuna who do the same sort of migration from the Western Pacific basin. So off the coast of Japan, traversing eastward to the subtropical North Pacific, and they have their own suites of models to kind of target that subpopulation of these fish. So there's a bit of a caveat there that this is, really targeted to a specific region. But I think with the sample set that we have, with multiple fish for every sub-migratory class, and just it's amazing looking at some of the 2 or 3 year long tracks, and they are doing the same exact swim every year. They're very consistent about where they're going, the timing of their migrations, and even across fish as well. So, of course, would never say no to more data, but I think just given that this is a fairly important fishery for the US, these tagging programs have been around for quite some time. I think relatively in the world of species distribution models, this is about as good as we can do at the moment. But all ears for different technologies to get more information from the fish. But yeah, it feels like there has to be a better way than tagging individual fish and then hoping they swim back to shore or somebody catches them. So.
Is it on? Yeah. Thank you. That was really interesting, really great talk. I was just curious, you mentioned that, the predictor variables, that they were quite few, not that many. How did you choose the predictor variables? And are there others that you could use, but you just don't have the data, or how does that?
Totally. It was really interesting looking at a few other species distribution models, not only for albacore tuna, but other marine species. I would say selecting variables for these species distribution models is really dependent on what the end goal of the models are. So if I were trying to create the absolute most robust model to really understand where albacore tuna are going and not going, I've seen models with 20, 30 different predictor variables, but that was not really the goal of this study. It was really to try and isolate the impact of biogeochemistry on their movements, and that becomes a lot more clear the fewer the variables, trying to minimize correlation between variables. So I guess we had the most time spent selecting the proxies for optimal foraging conditions, the chlorophyll and POC. There were a few other variables that we could have included in the model, like backscatter, which all of those provide a slightly different perspective into biological productivity. But really the goal here was to use kind of the rawest form of data coming from the floats. So trying to minimize downstream processing. We could have used net primary productivity, but that involves multiple steps from the raw data that you get from the float all the way to, perhaps a more all-encompassing variable to represent foraging conditions. So yeah, spent a decent amount of time thinking through which variables it made sense to include for these foraging proxies. But other than that, the variable selection was really dictated by what we could get kind of directly from these BGC-Argo floats, because that was kind of the main kind of end goal focus with this study. So, but yeah, there are models that use many more variables than what we used here.
Andrew.
Thanks so much for the talk. That was really interesting. You showed that places where the float model accurately predicted species presence, but the satellite model didn't. And I was wondering if you looked at the reverse and if there were any spatial coherence to places where the satellite got presence right, but maybe the float model didn't.
Yes, that's a really good question. It was definitely less coherent for the satellite model than the float model. I guess on the and the other kind of interesting tidbit from that comparison, I guess that was another example. There was another example where the float model performed much better in predicting presences than the satellite model. And that was kind of off the coast of the California current, kind of beyond the upwelling front a little bit. That that kind of creates a like very dynamic frontal zone where surface satellite tends to be, or surface chlorophyll tends to be quite low, but just because of this like kind of intense physical processes and mixing happening below the surface, there tends to be a lot of fish presences here, or like in that region. And that was another area where the float model really outperformed the satellite model in identifying presences. So it's kind of a multi-step process in first figuring out like, okay, where are those coherent regions, as you were saying, and then like, what are some, is that supported by like diving behavior that we see from the fish there, and are there oceanographic processes that can also help to explain why one model might be outperforming the other. I can't think off the top of my head of like coherent places where the satellite model outperformed the float model. I'll go back and check now.
I want to ask one question before we close. The Bioargo floats are quite sparse, right? And then it's a machine learning approach run mostly in the Copernicus system to make these estimates of the 3D varying fields. They have their uncertainties, and then the uncertainties of your approach. So how much do those uncertainties propagate? It seems pretty clear that it's an improvement, even if there is uncertainty in those 3D fields. But so that's sort of one question. And then I guess the other question I have is how much of this is just like climatological improvement as opposed to truly interannual varying improvement?
Yes, really good question. Yeah, so for first question, all of those uncertainties very much so exist. And at least from kind of some of the comparisons that I have done, I mean, people have used like modeled variables for all of these, for some of these biogeochemical variables in the past. And at least from some comparisons that I've looked at, these data products capture like spatial and temporal variability so much better than some of these biogeochemical models do. So that I think is a true marked improvement. But I like, even these data products are still only monthly resolution. So there's only so far we can zoom in and also kind of trust that temporal variability. That was actually like a fairly big challenge with this project. Like, this kind of monthly resolution was about as good as we could do for some of these biogeochemical data products, but the fish data has like a minute resolution for where the fish are, with some caveats like, there are error bars on where the fish spatially is located and how deep that they're diving, but there's still a pretty big mismatch in temporal and spatial scale for the fisheries data and the biogeochemical data. So definitely movement to be done still on that front. And I think there's a lot of variability that we're missing in terms of time, just having monthly resolution as opposed to, the minute resolution that we have of the fish. So we're getting there. This is an improvement, but work to be done for sure.
Yeah, great. Well, we have a question on
and seeds.
Let's see. I was wondering how you went about selecting what type of fish species to study. Was the albacore tuna selected for its geographic significance, or
One of our collaborators, it's not a super interesting answer. One of our collaborators is from the Southwest Fishery Science Center at NOAA, and she is an albacore tuna specialist. And again, part of the reason that we chose this species was because we have access to this pretty robust data set of archival tags. So it really came down to a fish of opportunity, I would say.
all right, well, let's thank Mary Margaret again. Thanks everyone for coming.